Hi everyone, and welcome to our summary video all about the ultrastructure of eukaryotic cells. This time we're going to focus on those organelles without membranes. So this relates to a couple of references in our specification. First of all, having a look at the ultrastructure of those eukaryotic cells and the functions of the cellular components with a focus on our ribosome, centriole and the cell wall. And also the importance of the cytoskeleton, so looking at how it provides mechanical strength, aids the transport and enables cell movement. So our first structure that we're going to look at then are the ribosomes. Now when we are thinking about the ribosomes, we probably heard about these at GCSE as the site of protein synthesis. What we need to know for A level is a little bit more. We need to know a little bit about their structure and of course their function. So if we think about structure first of all, then what we can see is that they are these small spherical organelles. Now they are small, we're looking at about 20 nanometers in diameter. As the title of this little summary suggests, they are not bound by a membrane. They're made of two key components, if you like. They're made of protein, and they're made of this stuff called rRNA. Now that little lowercase r at the beginning just stands for ribosomal. So what we actually have is ribosomal RNA, or the rRNA, and the protein make up our actual ribosomes themselves. Now what we have are actually two subunits. Now those two subunits are going to be made in the nucleolus, and then they're assembled in the cytoplasm. So what we see happening is within the nucleolus, where we obviously find the RNA, then we're going to make those two individual subunits, the large one and the small one. They then get transported to the cytoplasm, where they're then put together to make the overall ribosome structure. Now, in terms of where we're going to find these ribosomes, a couple of options. Firstly, they could just be free within the cytoplasm, not attached to anything, that means. Or they could be attached to the endoplasmic reticulum and therefore obviously the rough endoplasmic reticulum, because if we think back to our previous summary about the membrane bound organelles, endoplasmic reticulum has both rough and smooth. Smooth has no ribosomes on the surface, the rough has the ribosomes on the surface. So what do? Well, if we think about our ribosomes on the rough endoplasmic reticulum, they are going to synthesize the proteins that are then going to be exported from the cell. So basically, any of those ribosomes on our RER, they're going to be making those proteins that we're then going to transport out of the cell. Those ribosomes that we find free within the cytoplasm, these are still going to assemble proteins because that is what a ribosome does, but those are going to be proteins that are used within the cell itself. So two different places that we find our ribosomes, both of them making proteins. Rough endoplasmic reticulum ones, they're going to export those proteins from the cell, whereas the free ribosomes, they're the ones that are going to be remaining within the cell. Our second structure of this summary are the centrioles. Now, the centrioles themselves are actually made of these two bundles of microtubules that are arranged at right angles to each other. And you can see that in the little diagram down the bottom here. So you can see that we've got one bundle of microtubules running kind of towards the right of the screen, and then the other bundle of microtubules at 90 degrees to them, so at right angles. When we look at those microtubules, they're made of that substance called tubulin, and they've been arranged to form that little cylinder. And you can see it in the diagram there that all those little microtubules made of the tubulin are just arranged in that cylinder shape. And then two cylinders at 90 degrees to each other, that's what we're referring to with a centriole. So what do the centrioles do? Well, they've got kind of two key roles, if you like. So the first role is looking at cell division. Now, in terms of cell division, these are the ones that will form those spindle fibres that hopefully we heard about at GCSE. And these spindle fibres are going to come out when we're looking at those chromosomes being separated in mitosis-meiosis. Now, what happens is the chromosomes attach to the spindle fibres, and then, previously, what we said was they magically move to the poles of the cells. Obviously, we're going to break down the magic at A level. We're going to understand exactly why. So once the chromosomes have attached those spindle fibres, then we've got these little motor proteins that literally move along those tubulin fibres and therefore move the chromosomes to the opposite poles as they do so. So it's kind of like using those tubulin fibres, those spindle fibres, as pretty much like a track, and the little motor proteins are the ones that are then just going to move along the track, taking the chromosomes with it. Second option of what we're using them for is forming our cilia and the undulipodia. Now, hopefully we remember the cilia are located within things like your respiratory system, for example, and they are going to be very important in obviously moving things like mucus around. To form those cilia, the centrioles are going to multiply. They then arrange themselves just beneath the cell surface membrane, and then microtubules form from the centriole and the membrane is stretched around them. And if we remember, when we think about what those cilia look like, they're all these little tiny hair-like projections from the surface of the cell. So if you imagine that what's happened is just underneath that cell surface membrane, we've got our little centrioles multiplying, arranging themselves just under there. What they're then going to do is form these microtubules that are going to project out and take that membrane around them as they do so. So they're our little cilia. The third structure we need to know about is the cell wall. Now, we've heard of cell walls previously where we've looked at our plant cells. And hopefully we do remember that when talking about plant cells, the cell wall is made from cellulose. In animal cells, there is no cell wall, so it's made of nothing because cell walls do not exist in animal cells. One other cell wall we do need to know about are those in fungal cells. Now, fungal cell walls contain a different substance. They contain chitin rather than cellulose. So plant cell walls made of cellulose, fungal cell walls made of chitin. In terms of what the cell wall actually does, hasn't really changed much from GCSE. It's going to provide strength and support. It's going to help maintain the shape of the cell, and it is permeable to certain substances. We then come on to the cytoskeleton. Now, the cytoskeleton is something that obviously previously, lower down the school, we just ignored. We just sort of said, oh yeah, there's this jelly-like stuff called cytoplasm, and that's it in that area. It was a lie. We also have this cytoskeleton. Okay, now the cytoskeleton itself is actually relatively complex as a structure because it's made of a range of different parts. So what we have, these actin microfilaments, which are about 7 nanometers in diameter. We've got the intermediate filaments, 10 nanometers in diameter. We've got tubulin microtubules, 18 to 30 nanometers in diameter. And then we've got a range of motor proteins. So myosins, kinesins, dyneins, all of these are motor proteins. So what we have are kind of like those 4 key parts, if you like, that make up our cytoskeleton. Actin, intermediates, the tubulin microtubules, and then the motor proteins. What we're going to do is we're going to go through each of those individual parts of the cytoskeleton to understand what their actual function is, because they all do something ever so slightly different to one another. So they all have their own role to play within the cytoskeleton itself. If we think of the microfilaments first of all, so the actin microfilaments here, 7 nanometers in diameter, these are the ones that are going to provide support and mechanical strength for the cell. Can we please get into the habit of using this phrase mechanical strength rather than just strength? It's one of those examples where we're going to upgrade the language that we're using at A level to ensure that we're explaining things properly and not doing this half-hearted attempt at biology. So provide support and mechanical strength for the cell, and they allow for cell movement. Next up, the intermediate filaments. These are the ones that are 10 nanometers in diameter. They're going to be used to anchor the nucleus. So the nucleus isn't just free floating however where it fancies in the cell. It's actually held in place by these intermediate filaments within our cytoskeleton. We also find these intermediate filaments are able to extend between cells in certain tissues, and that means that those individual cells now have a method of communication. They can use this cell-to-cell signaling. So this gives them a communication network within that tissue itself. We also find that that extension between those cells in certain tissues allows it to kind of anchor down these tissues onto the basement membrane. So what it does is it helps to kind of stabilize the whole tissue through that process of adhesion to the basement membrane there. The microtubules then, the 18 to 30 nanometer ones made of tubulin, these are providing shape and support to the cells. They are also very important in allowing the organelles to move within the cell cytoplasm. The way that they do that, just like we saw previously with tubulin, what the tubulin does is it makes the network along which the motor proteins can move. So really, wherever you see this whole idea of tubulin, start thinking about motor proteins moving along it. So in this case, it's going to be the motor proteins moving organelles around the cell by following these tubulin microtubules. We already mentioned one of those other uses, forming the spindle during cell division, and the cilia and the undulopodia. So what we actually have are a range of different uses of our microtubules there. So just to finish off, if we think about the kinds of questions that can come up, particularly around the cytoskeleton, then one of the old favourites is to just state kind of 2 or 3 functions of the cytoskeleton. So what we need to be careful of here is that we're using the right terminology. So obviously providing mechanical strength, that's going to be one of them. Allowing organelles to move within the cell is another. Make sure that you are being that little bit more specific when you give those answers. So obviously we've gone through a range of different functions through those different parts of our cytoskeleton. Just make sure that you are specific when you give those answers, because as I say, if you're just to put down strength, it's not enough. So make sure you use that mechanical strength, make sure you are talking about the fact that it's allowing organelles to move within the cell, not implying that it's moving elsewhere, like outside the cell, etc. And then you should be able to score very well on those questions. So hopefully at the end of this video, you now have a greater understanding of those non-membrane bound organelles within our eukaryotic cells. And as always, don't forget to subscribe to the channel so you can see when new biology A-level content is uploaded.